in. Okay. Hello, everyone, and thank you so much for joining us for the August edition of the GSU Malaria Journal Club monthly series. Happy August. And I think we are a day away from Mosquito Day, which is tomorrow. So I think this Journal Club is quite timeous in that as well, that there's an important day where malaria is going to be discussed and other diseases as well that are caused and spread by mosquitoes. Today's webinar is a parasite-focused one, which is going to reference Dr. Balotin Fogang's recent paper titled Detection of Novel Plasmodium falciparum haplotypes under Treatment Pressure in Pediatric Severe Malaria. My name is Paolo Chauke, for those of you that do not know me, and I'm a training coordinator at the Wellcome Center Institute Malaria Gen GSU, working with a team of awesome people, some of whom are in the room right now, to organize these monthly events. So thank you so much to all of you for joining us once more. Just a few ground rules. The Journal Club is being recorded and will be available on the Malaria Gen YouTube channel later today. So you can rewatch if you missed something, or you can share with your friends and colleagues and networks as well to spread the message and knowledge that we're going to be getting today. Please don't mute so as to not to disturb Dr. Balota as he presents and speaks. We will have a Q&A session at the end where you can either raise your hand and I will give you the powers to speak, or you can type your question in the chat or Q&A box as well, and I'll read them out to Dr. Fogang so that we get a response as well. Without further ado, I would like to introduce our speaker for today, Dr. Balotin Fogang, who kindly agreed to give this presentation when I invited him a few months ago. So I'm really grateful and excited for this Journal Club as well. The title of the talk is going to be based similar to the paper as well. And just a short bio to let you know who Dr. Balota is. Dr. Balotin Fogang is a postdoctoral fellow in the GATAC Malaria Team Laboratory of Host Pathogen Immunity at the University of Montpellier in France. His research interests include investigating genomic as well as transcriptomic Plasmodium falciparum factors that predict malaria infection outcomes. Dr. Balota, thank you so much for joining us. You may begin your presentation.
Thank you for the invitation as speaker to this session of Malaria Genomic Epidemiology Journal Club. So I will first thank all the Malaria Gen community for joining this talk today. I will also give a special thanks to all the co-authors of this paper that I will share with you today. This paper entitled Detection of Novel Plasmodium falciparum haplotype under Treatment Pressure in Pediatric Severe Malaria. This work will be presented to you in two main parts. First, the context as the objective of the study, and then the result and the discussion. As we all know, hoping that most of you are familiar with malaria, Plasmodium infection may lead to three main clinical outcomes: the asymptomatic malaria, infected without any malaria-associated symptoms, to uncomplicated or severe malaria cases. And most of the death in malaria is associated with children under 5 years old, mostly due to their low acquired immunity to malaria. Since 2011, the WHO are defined as a front line for the treatment for severe malaria, the intravenous injection of artesunate. But in case there is not artesunate or artesunate is unavailable, the alternative is intramuscular injection of artemether or intravenous injection of quinine. ACTs have been recommended at the frontline treatment of severe malaria, both severe or for malaria, uncomplicated or severe malaria. But recently, the resistance to ACT have been risen in Asia and in some few countries in Africa. PFK13, that is known to be associated to the resistance to malaria, is the strongest predictor of in vivo parasite clearance rate after treatment with ACT derivative. But the frequency of PFK13 that is associated with resistance to ACTs is found a very low frequency in Africa. Recent studies have shown that has shown that by analyzing more than 6,000 falciparum patients treated with artemisinin derivative, they've shown that both in area with non-resistant of artemisinin and the area with artemisinin resistant, there was high variability in parasite clearance half-life in the both area. This raises to the question, why may be the factor that may explain the difference in the parasite clearance in the area with non-evidence of artemisinin resistance. Several factors may explain this high variability in parasite clearance half-life in the area with non-evidence of artemisinin resistance. This factor may include the age of the host, the host-acquired immunity, the initial parasite level before the treatment, the development stage of parasite before the treatment, or the genetic complexity of the P. falciparum before the anti-malaria treatment. And in severe malaria, also the sequestration of infected red blood cells in the profound organs. This question lead us to the aim of this study. That was to evaluate the association between the parasite clearance rate and the multiplicity of the infection in children with severe malaria undergo treatment, using a variety of sequencing methods and biological material. More specifically, in this study, we wanted first to evaluate the different methods to study the intra-host dynamic of polyclonal infection during the parasite clearance under anti-malaria treatment, and then use this method to determine the association between the parasite clearance rate following artesunate or quinine treatment and genetic complexity of Plasmodium falciparum in children with severe malaria. We carry out this study in Benin. It known that for now, there is no evidence of artemisinin resistance in Benin since the introduction of ACT in 2005. As the study design, we include in this study 65 severe malaria individuals, a part treated with quinine, and another part treated with artesunate. After arrival at the hospital, when the kits were included in the study, one blood sample were collected before treatment, and 3 additional blood samples were collected at 8, 16, or 24 hours post-treatment. All these samples were suggested for whole genome sequencing, and then we used 3 different metrics to determine the clonality of the infection, which allow us to classify our individual as monoclonal individual or polyclonal individual. Polyclonal here is meaning polyclonal at least a one time point during the 24-hour follow-up. And then all the polyclonal individual were suggested for, all the samples from polyclonal individual were suggested for amplicon sequencing using both genomic DNA from plasma and pellet. I will explain why later. The first aim of this study was to determine the multiplicity of the infection using whole genome sequencing. Sequencing and variant calling were performed by Malaria Gen, and as the sample of this project is the part of Malaria Gen P. falciparum community project. After the filtration of high quality sample, we used 3 different algorithms to determine the multiplicity of the infection. The FWS, which measure the intra-host heterozygosity in relation with population heterozygosity. The Rey-McCoy, which is a statistical framework that uses Markov chain Monte Carlo approach to determine the frequency of this distinct genotype in one sample. And the Longhorn of homozygosity will measure the relationship between 2 or multiple strains in one isolate, and based off the pattern of non-referent allele frequency, we can classify an infection as dominant clone, 1 dominant clone, 2 dominant clone, or more than 2 dominant clone. Based on this 3 metric, we observed that the reg marker was less sensitive for determining multiclonal infection compared to the FWS and the ROH. However, there was a robust correlation between the 3 different metrics. This allows us to detect 35 individuals and classify them as polyclonal infection. Here, a monoclonal infection was classified as monoclonal detected by the 3 different algorithms. After defining our individual as monoclonal or polyclonal, all the polyclonal infections were suggested for amplicon sequencing using both genomic DNA from pellet and from plasma. The rationale behind using these two biological materials was that red blood cell pellets may represent the circulating parasite at the time where we sampling the individual, while plasma could contain the parasite from the previous cycle that is independent of the sequestration, as we know that the mature stage of parasite, falciparum parasite, as sequestered. For amplicon sequencing, we target 3 highly polymorphic marker, the CPMP, the CPP, and the MO1. And we use the haplotype R package to reconstruct the haplotype. Here we have to notice that after the reconstruction of haplotype, it's always good to do an additional manual check to check some chimera that was not removed during the construction of the haplotype, because it's known that amplicon sequencing generates a lot of chimeras. Concerning the diversity of Plasmodium falciparum, we identified 65 microhaplotypes and 64 were found in the affected red blood cell, while only 7% were found in the plasma. In addition, the mean MOI was significantly higher in the infected red blood cell compared to plasma, indicating that genotyping parasite using the infected red blood cell pellet is better than using the plasma and the DNA source. However, at the high parasitemia, more than 1,000 parasite per microliter, more than 85% of our sample have the concordant result between plasma and pellet, meaning that at the high parasitemia, plasma can be used to effectively genotype parasite. This can be used in the retrospective studies. Altogether, this shows that amplicon sequencing using red blood cell pellet and genomic as source of genomic DNA is more sensitive compared to plasma at low parasitemia. We don't compare the amplicon sequencing with the whole genome sequencing based method. We observed that amplicon sequencing was correlated with the MOI. The MOI detected by amplicon sequencing was correlated to the MOI detected by FWS or ROH. But the amplicon sequencing was able to detect more than 6 genotypes compared to the RegMark call that is also a quantitative method. In addition, amplicon sequencing is the only method that can quantify the proportion of each clone in an isolate. This shows that combining different haplotypic techniques can be used to characterize the genetic complexity quantitatively and qualitatively within an isolate. Having established the method that we can use to determine the multiplicity of the infection, we now want to determine if this multiplicity of infection is associated with the parasite clearance rate. We observe, as expected, that individuals treated with artesunate clear faster the infection compared to those treated with quinine, because artesunate is a fast-acting drug. However, I observed, before we observed the high variability in parasite clearance rate in both individuals treated with artesunate or quinine, varying from 4 hours to 24 hours. This shows that there is a complex dynamic of parasite response to treatment in the early stage of infection management. We took the advantage of time series sample to determine the parasite clearance rate, the parasite clearance pattern in each individual, and we observed a rebound in parasitemia in more than 30% of the individual. The rebound here is defined as the increase of parasitemia in more than 1,000 parasites per microliter between 2 time points. This rebound in parasitemia can be explained by the fact that the potential release of the sequestered parasite during the malaria treatment. To determine if the multiplicity of the infection was associated with the clonality at the start of the treatment, we compare the parasite clearance pattern between monoclonal and polyclonal infection at the start of the treatment. In individuals treated with artesunate, there was no difference in the pattern of parasite clearance in both monoclonal and polyclonal individuals. However, in individuals treated with quinine, we observed that individuals with polyclonal infection clear faster the infection compared to those with monoclonal infection, although non-significant. But we're thinking that this low clearance of parasite in the monoclonal infection was due to the high parasitemia at the start of the treatment. So based on this, we conclude here that there was no association between parasite clearance rate and the multiplicity of the infection. Despite the high decrease in parasitemia during the 24-hour post-treatment, we observed that there was no variability in the mean MOI during the 24-hour post-treatment. This could be explained by the fact that we detect the new microhaplotype after the start of the treatment in more than 60% of our sample. And also, half of the individuals with monoclonal infection at the start of the treatment have the sporadic microhaplotype during the 24-hour post-treatment. This goes in line with the fact that there is liberation of some sequestered parasite after the start of the treatment. And this was no association with the rebound in total parasitemia that we observed before. Altogether, this shows that it's usually not possible to genotype all parasites to microhaplotype from just one blood sample collected in one individual at the arrival at the hospital. We also determine the allele frequency of different resistant marker in individual during the treatment period. And we observed that there was no alteration in genotype across all resistant markers within the same individual during the active malaria treatment, meaning that there is no association between the parasite clearance rate and the different resistant marker. In summary, in this study, we show that combining different haplotyping techniques can effectively determine the parasite genotypic complexity, and also plasma can be effectively used for parasite genotyping, but at the high parasitemia level. This can be used maybe in the retrospective study. And also, we show that parasite clearance rate in severe malaria individual is independent of the multiplicity of the infection. And then genotyping a blood sample from a patient at the arrival at the hospital is typically not sufficient to capture all the full spectrum of parasite genotype present in an infection. So we need more sample time points to be able to genotype all the microhaplotype from Plasmodium falciparum in an infection. From this, I will thank all the co-authors and all those who contribute to this study, and also the funding sources and other support. Thank you for your attention.
Hey, thank you so much, Dr. Balota. This was fantastic. We have time for questions now. I see there's lots of questions already in the Q&A, which I appreciate, and I'm going to encourage people to ask more because I think they're from one individual, which I'll read now. I'm going to take the honor of being the host and ask you questions first. So yeah. I have about two. I have a lot, but I chose two from those. So I think for me, what stood out when I read your paper was the sampling time frame. I think you sampled patients every 8 hours over a 24-hour period window after treatment onset. So my question is, why that time specifically? Why not include later time frames like 32 hours, 48 hours, or 72 hours? Why specifically a day or 24 hours? As there could be perhaps delayed haplotypes or emerging differences as well that could be discovered, I think. And then the second question is, does this have anything to do with pharmacogenetics? Because I think you focus on the complexity of the genetics of P. falciparum, but what about the children or the hosts themselves? Could there be something to do with how the drugs are interacting with the people's genes? Thank you so much, Dr. Balota.
Thank you. The first question for why just 24-hour? Because here we wanted really to monitor the haplotype that was present at the beginning of the infection during the 20, is why we did not go really further. And most of the children are also, we have also a technical issue. There was two problems. There was first the technical issue because some of the children, sorry for them, they was at certain point drop out of the study of either because of death and other we lose them. It's why most of the time we decided to stop at 24-hour. There was some sample that we have more than the goal was 24 after 24, 7 days and 32 days, but we did not have enough sample after the 24-hour. So that's mostly sometimes technical issue. The second question, I don't remember the second question, but please can you repeat the second question?
Sorry, yes, thanks, Dr. Balota. So the second question was about pharmacogenetics of the
Yeah.
hosts. Because then I think you focused more on the complexity of the genetics of P. falciparum, but not the children themselves, as though how the two drugs that were administered interact with them differently due to their genetic makeup.
Yeah, to be honest, we did not really check about the pharmacogenetic of the drug in the children. We mostly focus on the parasite aspect.
Okay, no, thank you so much for engaging me on my two questions.
But for the whole project, there was the other aspect about the host genetic aspect. So I was no one in taking part of this part. The host and immunity part of the project, I was not the one taking care of that part of the project.
Thank you so much, Dr. Balota. We have time for questions, so please do raise your hand. You are encouraged to raise your hand or type in the Q&A or chat box. I see there's lots of questions, so I'm going to start reading them out. Apologies to Dr. Foucault. I see you have like about 5 questions. I'm going to start with the individuals with 1 question each and then come up to you afterwards. Yeah. I'm going to start with Chun Lee, who says, in the journal club description, it says you work on transcriptomic factors. Are you working on any extensions of this project using transcriptome data or in other projects?
Yeah, for this project, there's a transcriptome part and there's also some paper published about the transcriptome part for the same project. But for me, for this project, I just focus on this part of the project. This is not really my main project. In my main project, I really focus on transcriptome and genomic factor that can predict with the outcome on an infection. We have different cohort from different countries in Africa, Cameroon, Ghana, where we collect samples during 3 months, and then we are looking about the transcriptome and the genomic aspect, if it can predict the outcome, be asymptomatic or symptomatic. So.
Thank you so much for that, Dr. Balota. There's an anonymous attendee who's asking, can we get the recording of this presentation? Yes, anonymous. We are recording this Journal Club. You can find it on the Malaria Gen YouTube channel later today. And you can find old recordings as well. Adwa Agiopama says, can I confirm that high parasitemia was defined as 1,000 parasites of blood? That's from Adwa.
High parasitemia, that's we did not really define high parasitemia here. So most of the sample was more than 10,000. In severe malaria individual was more than 10,000 parasite individual. But here, high parasitemia, we did not group our sample as high parasitemia or not. It's just when we analyzed the amplicon sequencing between plasma and pellet that we realized that at the parasitemia above 1,000 parasite per microliter, the result can be concordant between the both biological material. We just wanted to show that if you have high parasitemia in an individual, you can use plasma to effectively genotype parasite. This is mostly used in the retrospective study. But according to WHO in severe malaria individual, high parasitemia is 10,000 if remember was 10,000 parasite per microbe.
Okay, thank you so much, Dr. Balota Fogang. I'm going to read a few comments and questions from Dr. Loic Foko, who's been very engaged, and we're grateful for that. So I think the first one is a comment, and then the second one is a question. So the first one is from Dr. Foko. Some of your correlation values between the three techniques are below 0.80, which suggest a mitigated correlation between them. And then the question is, have you tried to stratify the proportions of isolates detected based on each method as per parasitemia threshold, low parasitemia versus high parasitemia? Dr. Fogan?
The thing is that most of in terms of whole genome sequencing base, it was the same genome that we analyzed with the same sequencing that we analyzed with the three different methods. So I think we can directly compare because it's exactly the same sample that you compare with three different methods, same sequencing, but we did not really stratify in low parasitemia or high parasitemia. But I think it's comparable between the three whole genome sequencing approach.
Okay, thank you so much for that. Another question from I'm gonna read a comment and question from Dr. Foko and then move to other participants as well and come back to another question from Dr. Foko. So this is the comment, I think. Your standard deviations for mean parasitemia at each sampling time are very high. It might explain the absence of significant association between multiplicity of infection and parasitemia in the two drugs that you were working on. The question is, how can your findings be translated into policy or a guideline? That's from Dr. Foko.
Yeah, we noticed that the parasitemia in quinine and in individual treated with quinine was really higher compared to those treated with artesunate. So for now we don't have any explanation why, but it was from different years because it was 2014 for quinine groups and 2016 artesunate group. So I don't exactly know why and how. Why is this side is different?
Okay, thank you for that. There's a question from Moses Igbe Bunam. Are you recommending use of many techniques to determine multiplicity of infection? Is this cost effective for endemic regions?
I would not really recommend using multiplicity of multiple technique, but it will always depend of your question and what you want to analyze in your study. Because if you have time series study and your goal is to see if it's the same section throughout, you can just use the amplicon sequencing because you can easily see which genotype are here and which genotype the next day and which genotype the third day. But this is only to see the multiplicity of the infection. You can use whole genome sequencing or other sequencing approach based on SNPs. So it's depend of I would say it depends of which question you want to reply. What is the goal of your study? So
thank you so much, Dr. Fogan. There's more questions coming in, which is making me happy. We have a question from Dr. Richard Pearson, who we have multiple questions from Dr. Richard Pearson, who is the parasites surveillance lead at Malaria Gen. I'm going to read the first one, which says, I think your earlier slide suggested that longer clearance times were seen in artemisinin sensitive areas. Is that correct? If so, this is surprising, as I would have expected longer clearance times in artemisinin resistant areas. Dr. Fogan?
yeah, according to the study from White in 2019, he showed that there was no really a huge difference between the both area because he analyzed more than 6,000 samples from P infected individual, P falciparum infected individual treated with different artemisinin derivative, and he saw that the high variation was observed in both area with non-resistant and area with resistant. Even us, we was expecting that. yeah, it's what is shown in the literature. So.
Okay, thank you for that, Dr. Balota. So I'm going to go back to the last question that was asked by Dr. Foko. What were the main clinical phenotype of the severe malaria cases? Was it SMA? Was it ARDS, etc.?
I think we have severe malaria, we have severe anemia. I don't remember the proportion, but it was different. Okay. Oh, I don't remember the proportion, but we have severe malaria, severe anemia, and others. So I don't remember exactly the proportion, but we have the different groups: severe, those with only severe anemia, those with severe malaria, those with cerebral malaria, and severe non-cerebral malaria. So it was a mix of. But when we try to categorize them by groups, we do not see any association. It's why we decided to combine all the groups in just one group.
Thank you so much, Doctor. So there's a question from Mariscal Bryce. Is it possible to translate these findings carried out in Benin to other African countries?
Yeah, of course. I don't see any. Normally, it should be possible. If you can have a cohort, or normally you can analyze the multiplicity of the infection for any sample that I think it's possible.
Okay. Thanks for that, Dr. Balota. We have two questions from Dr. Andrew Barmer. So I'm going to read them one by one. So I'm going to ask one and then let you answer and then read the second one. So Dr. Andrew Barmer says, thanks for the very interesting talk. I had two questions. The first one, how repeatable were the measurements in parasite density? If you measured the same patient at the same point several times, would you see a lot of variation in the estimate of density?
Generally, because here it was microscopy, generally it's two independent microscopy. The cell was read by two independent microscopists. And then if the result, one result was discordant, there was a third microscopy with that slide. So I think based on that, we trust the results.
Second question from Dr. Barmer. Could you go into a little more detail about which other factors might affect parasite clearance rates? Why do you think you see so much variation? Did clearance rate vary by parasite background?
Yeah, I think there could be many factors associated with that because I've shown we think that, for example, in our study, we showed that in polyclonal infection clear faster, but we think that is not associated with clonality, but it's associated with parasite levels. So if the parasitemia is higher at the beginning, we expected that the time for clearance could be higher than the ones with low parasitemia. You can also have the host-acquired immunity because the parasite clearance is not just based on the drug, but it's also depend on how the organism, the host human organism can react to help parasite to be clear faster. So you have host-acquired immunity, and you can also have, it's also depend on the stage of the parasite because if you take an individual when they are, it's only the mature stage, those stages will be sequestered. So they will take some time to be released in the circulation for drug to act to them. So the clearance time will be longer than if it was at the ring stage when you treat the individuals. So there's several factors that could explain this, but it's really difficult to capture all those factors at the time. Sometimes you can choose just some factor and try to analyze them. It's difficult to capture all those factors at the same time.
Okay, thank you so much for that. Just to say, you are allowed to raise your hand and ask a question or give your comment. However, I'm happy to continue reading your questions in the Q&A box. Keep them coming. I'm happy that you're also engaged as well. I think you answered this earlier, but I'm going to ask it again. It's from Chun Lee, who says, more on transcriptomics. What genes are you interested in when looking at a transcriptome data?
Actually, my team is mostly the GATAC team is mostly focused in version. So what we generally do, we have a lot of sequencing approach. So when we're looking version, we first do the GBL alpha. Is more consist domain in version. So to look if the version expression can be associated with the outcome, we sequence the version from an early infection time point and see if it can be associated with the clinical outcome. And then from that, if you have the sequencing of DB alpha, you can predict the full length version using VARIA on other software. And also we do the bulk RNA-seq. So bulk RNA-seq, you have the whole picture of transcriptome in your sample. And then for some specific sample, we also do the single cell RNA-seq because sometimes we have like a mixed infection or a mixed with genotype or parasite at different age. So you have to do single cell to be able to separate the different parasites to see if there's any association. So, and then we do a lot of there's different approach depending on which question you want to apply. You can sequence parasite depending on.
Okay, thanks, Dr. Fogan. We have another question from Dr. Pearson. Why do you think you are seeing greater complexity of infection in amplicon sequencing data than in whole genome sequencing data? Do you think this can be explained by greater sequencing depth, or might there be other reasons?
Yeah, we know that in general amplicon sequencing is any gen is more sensitive than using the whole genome sequencing because it's true that the Malaria approach is the selective whole genome amplification. I don't know it could depend on the depth. But in general, it's known that amplicon sequencing is really sensitive because you just target the portion of a gene. It's easy to amplify just 400 or 380 base pairs that amplify the whole genome. So it's why amplicon sequencing, and then you do multiple cycle of PCR. So normally it should be highly sensitive than the whole genome sequencing based approach.
Okay. We have a question from Ankashka Jarav, who says, in the presentation, the first sample was collected before 0 hours, as in before the admission of the patient and later after 4 hours. Why was it collected before the admission when the parasite was already present and probably the infection could still be high?
The goal of collecting before the treatment was to have the parasite genotype before any action of the drugs. To see, because if you want to see if something happened when you treat the individual, you need the starting time point to be able to know if this affect what happened later. If the drug, yeah.
Okay, so Mariscal is back again to clarify. So I think Mariscal Bryce asked earlier if the studies from Benin can be translated into other African countries. So I think they're clarifying here. Mariscal says, I was asking if the high standard deviation could explain by the long-term cryopreservation 2014 and 2016 till now, which can induce parasite adaptation as well as genetic modification. Any comments on that?
I don't think so. I'm thinking that even in the same cohort, the standard deviation is already high between patients. You have patient with 1 million, you have patient with 10,000. Both patients have severe malaria. So I just think that I don't think that is really dependent to the fitness course of the adaptation of the parasite during the different time point. Because in individual, in sampling in the field, the parastemia is always very highly variable between individuals. So it could be dependent on many other factors. I don't think that it's
Okay. So another question from Chun Li is, sorry if I missed this, which amplicon panel are you using?
We target three highly polymorphic genes, the CP, MP, the CPP, and the MA1.
Okay, thank you so much. Another question from Dr. Pearson. Do you have any suggestions of what approaches should be used for global genomic surveillance of Plasmodium falciparum? For example, strengths as well as weaknesses of amplicon sequencing versus whole genome sequencing. Is the added value of collecting multiple points justified by the increased cost, etc.? That's from Dr. Pearson.
For highly approach, it depends because if amplicon sequencing, as I will show, it really depends of the goal of the study. But this is the global health approach. I think amplicon sequencing is not the best. But there's other many methods. It's not only whole genome sequencing that you have now. You have some method that MAT4, harder, or that is used now. So you have some other methods for that. But amplicon sequencing is mostly. I will use amplicon sequencing mostly in a cord, because in cord you can easily, if you want to follow the trend of your parasite changing, you can easily see from one time point to another time point if it's the same parasite or if the proportion have changed or not. But if it's just it's a global approach, I think it's better to use something based on this the SNP base, something based on the SNP, because you can easily characterize different population based on the SNP structure. Because from one country or so one area to another one, the variation is really high. You need something that have technically the full spectrum of the genome, like the SNP based approaches. I think it's better in this case.
Okay. Thank you so much, Doc. There's a question from Becca Raya Abagero. How many genes are used in your macrohaplotypes to determine the multiplicity of infection?
Yeah, here we target three highly polymorphic genes. I said before, CPMP, CPP, and MA1. And then the haplotype was defined as the marker with the highly diversity compared to the other. Like in one sample, you have the sequencing of the three different gen, and then you choose the amplicon sequence, the genotype based on which one have the highly diversity compared to the other two markers.
Okay. And we have one last question, but we still have time, by the way. So I encourage you to ask questions, raise your hand or type in the chat box. We have about 15 minutes to go. There's another question from Chuan Lee. In your run of homozygosity analysis, do you generate plots of NRAF and the percent het or cause, then classify clonality manually, or do you have tools to do this?
Yeah, we have the tools that we modify from Pearson al in 2018. He was the one developing the Python script, so we just modify it, and then from that you can directly generate the plot with non-referent allele frequency. You can directly generate the plot that you see from my slide using that script, and then you need manual visualization to see if to see the pattern. Yeah, we did not develop something to be able to look the different pattern in each isolate. But yeah, we did not do that because we did not have so many samples. So for now on, we can easily go through all the samples to see, yeah, with 2 peaks, it's 3 peaks, or it was not that difficult to do that. But the whole script can directly generate the plots, and you can easily see from the plot to see if it's possible to do that.
Okay. We still have 14 minutes to go. I'm gonna let people raise their hands or type questions in the Q&A box. Going once. Going twice. Okay. Dr. Balota, any last comments, nuggets of knowledge of wisdom and knowledge from you before we close?
Not that much. I would just say thank you to all the community for joining the talk today. So thank you.
Okay, I will say this, quite impressed by how you filled up the questions. Fantastic presentation. And I think you've had one of the longest Q&As in our Journal Club series. So well done for answering so many questions so thoughtfully as well. Dr. Pearson says, great talk. Thank you, Balota. And I do want to end on that note to say, really, this was fantastic. Thank you so much, Dr. Balota. To all of you attendees of this Journal Club, thank you so much for joining us. We will have another Journal Club on the 30th of September, which is going to be a vector-focused one with Dr. Messimine from Cameroon. So I'm looking forward to that. Be on the lookout on the Malaria Gen LinkedIn, Blue Sky, and X pages, as well as a newsletter. Go to our website and sign up to our newsletter, and you'll receive the announcement in time. For those of you that don't know, tomorrow is World Mosquito Day, so happy World Mosquito Day, even though mosquitoes are destroying so many lives. I think most of you are interested in that, so hopefully you have a nice celebration tomorrow as well. But for those of you that are going to be joining us next month, see you on the 30th of September, and we are grateful for your support. I see there are other comments, which I'm going to read. Parath Lim says, great talk, thank you. Blessing Chamaka says, great presentation. Thank you, sir. Dr. Foko says, thanks, Dr. Balota, as well as the team. Truly, I want to end it on that note to say we are really grateful for your joining us on a monthly basis and to our presenters. So, Dr. Balota, you've made it onto the list of our Journal Club presenters, and you were
Thank you.
fantastic. Have a good day, everyone, and thank you so much. See you next month.
See you.